Hello, my name is Brandon Turner, and I'll be giving a sort of overview of different applications of probabilistic linking functions. This is a pre-recorded presentation for the 2020 Median Society for Mathematical Psychology. I want to start by saying that while there are many different ways of linking brain to behavior, this talk will focus exclusively on types of models that impose reciprocity, meaning that information from both streams of data are used simultaneously within the inference procedure. Perhaps the simplest approach is to specify a transformation of parameters from one stream of data to explain another. A common strategy in this framework is to simply regress parameters delta that describe characteristics of your neural measurements denoted in onto latent variables theta. The variables theta, having been specified through this transformation, are then deterministically related to the behavioral data denoted b. Another approach is to instead treat both parameter sets as random draws from an overarching distribution, such as the multivariate normal distribution, denoted by a set of parameters shown here by omega. Some new approaches from our lab instead place a structural constraint on the temporal properties of the measures, where here t is the designator that specifies how much variables should express covariance. A final approach is the integrated approach, which Cheng-Feng has already talked about within the symposium. One approach we've developed we refer to as joint modeling, where the joint here refers to the statistical distribution of neural and behavioral data. To build a joint model, we start by simply identifying some candidate brain regions, such as the striatum shown here. Ideally, the brain regions of interest will have some type of associated time series. This is, after all, their primary advantage over behavioral data alone. The time series should be tied to the experimental design somehow, where stimulus onset is shown as the red triangles here. We then have to develop ways of relating the neural activity to the stimulus design such that we can associate fluctuations in the time series directly to the stimuli. And suppose we do that with a set of parameters, and we're going to call them delta. In parallel, we develop some kind of computational model that is appropriate for the cognitive task at hand. The best form of linking comes with a model that also makes an explicit prediction for the time series information as a function of the stimulus design, just as what we specified in the neural stream. We'll again assume the set of parameters exists that give us some degree of flexibility in fitting to actual behavioral data, and we're going to call those parameters theta. Finally, we need to specify how the parameters delta and theta coexist. This can be something rather simple, such as the humble GLM, or it might be something multivariate, such as a multivariate normal distribution. In the end, the final specification of how changes in delta induce changes in theta is what creates a probabilistic version of the linking function. Here, the strategy is to simply specify that a link may exist, but the objective is to infer the strength of that linking function. And if we were able to fit a full model like this to data, it would give us a few advantages, perhaps the strongest of which is the induced reciprocity inherited from the hierarchical structure of the model. Specifically in this framework, the pattern of neural data can be used indirectly to constrain the parameters of the behavioral model and vice versa. By using the theoretical commitments of the cognitive model instantiates, the argument is that we are better postured to interpret highly complex patterns in neural data. So now I'm going to tell you about one application where we've used the covariance linking function to enhance the classic diffusion decision model's fits to behavioral data by connecting it directly to a time-evolving signal through brain data. For this application, I'll use the classic diffusion decision model. I'll avoid all the details of the model but stress one simple point. Like most decision models, the DDM comes with an explicit assumption of stationarity, or independent, identically distributed data. This means that the distribution of all behavioral measures is exactly the same for every trial. It doesn't matter if observers have some momentary frustration or lapse of attention, the predictions for the observer's choice at each moment in time is exactly the same. As you can imagine, this particular assumption is not well supported by real experimental data. Here's one example from a nice article showing some interesting patterns within RT data. The point is that there are clear fluctuations in the response time data that the off-the-shelf DDM wouldn't naturally predict. In other words, there are some aspects of the data that standard models are not prepared to explain from a theoretical perspective. So it begs the question then, how might these fluctuations be better explained? Another line of research tied to the same problem addresses whether or not behavioral measures can be meaningfully predicted by neural signals. One key observation is a systematic relationship of activation of the so-called default mode network, and how that relates to accuracy as well as response times. As one example, Icelli et al. Presented an analysis targeting the default mode network's activity leading up to an error trial. What they did was determine when errors occurred in their data, and then sorted trials on the basis of how far in the future an error would occur. They then examined the bold response as a function of this error lag and plotted the results shown here on the right side. As you can see, one network in the brain clearly decreases its average activation leading up to an error, whereas other areas in the brain systematically increase their average activation. Given that the brain can provide a natural covariate for decision models, the obvious question to ask is, can we somehow use the brain's trial-by-trial activity to predict someone's behavior? To address this question, we developed a joint model to link the neural data directly to parameters of the DDM. We start with the DDM's representation shown here. In a Bayesian framework, we can model the parameters of the DDM to fit choice response time data on each individual trial. For the neural data, we identified a large set of ROIs, extracted their time series information, and modeled the activity in each ROI as a function of the presented stimulus. We call the set of activations for each ROI on each trial delta. The last step was to assume a linking function to control how fluctuations in the brain manifested in fluctuations in the cognitive model parameters through the linking function omega. We then refer to this model as the NDDM, as it simply extends the basic architecture of the DDM to include neural covariation. Now going back to the problem of stationarity, we can examine whether fluctuations in the RT data are predicted by the NDDM. Here's a plot of the RT data from the experiment as a function of trial on the log scale. As you can see, there are potentially interesting fluctuations in the RT data. Now if we fit a standard model like DDM to these data, regardless of how well it fits summary statistics like the choice RT data, once you collapse across time, this plot reveals that the fits to the particular structure of RT observations leaves some aspect of the decision process that is still unexplained. When you fit the NDDM to these data and generate predictions, you get something much more noisy, but also pretty well correlated with the RT time series. To be fair, the NDDM has access to information that the DDM does not, but this is precisely why the focus on a joint characterization of both neural and behavioral data is a potentially exciting way to advance our understanding of cognition. Going back to the structure of the model, recall that I generically defined this linking function with a set of parameters omega. In the previous example, omega was actually a deterministic node in the graph and is comprised of a multivariate mean vector and a variance-covariance matrix sigma. In the previous analysis, I considered all possible pairwise combinations within sigma as a really generic way of building in highly multivariate brain coactivation patterns. But I can tell you this is very computationally costly, sometimes taking several days to fit the model. At the time, I already knew this would be a prohibitive cost when considering more realistic scenarios such as doing this analysis at the voxel rather than the ROI level. But we can still make use of the statistical structure of the model, it just need to place stronger constraints on the variance-covariance matrix. So for example, we can impose a factor analytic constraint to greatly reduce the number of parameters we need to estimate when fitting the model to data. So as you can see here, rather than estimating the full variance-covariance matrix, you can just estimate lambda, phi, and psi. This is a far sparser representation of the brain's coactivation. And I should also briefly mention that you can apply regularization methods such as the lasso to further improve the scalability of this approach. So the idea here is that if you have a very small loading from one brain region to a factor of interest, the lasso would push this estimate towards 0 while still allowing other loadings to remain unchanged. This promotes a much more parsimonious image of the brain behavior map to emerge all through structured prior impositions. Much like the connection across trials, the same basic covariance linking function structure can be used to connect multiple measurements simultaneously. In the next application, we use covariance linking function to connect behavior, EEG, and fMRI. So in another line of work, we asked whether there was some way to use the description of the decision process, that is the cognitive model, as a bridge to connect EEG, fMRI, and behavior. The rationale is clear. If all of these measures, EEG, fMRI, and behavior, are just different measurements of the same cognitive operations, then it makes sense that theoretical models of cognition can serve as the medium by which they can all be jointly explained. This is motivated by plenty of other studies using EEG to inform fMRI analysis or vice versa, and this line of research is often called data fusion. Here, the typical approach is to analyze these separate streams of data separately, and then perform some type of similarity analysis through correlation or representation similarity analysis. As is central to the theme of this talk, our research question was whether or not it was possible to jointly infer a common statistical structure in the data so that EEG could drive the analysis of fMRI and vice versa, all while testing the plausibility of our cognitive model. For this, we developed a three-way joint model. Conceptually, this isn't a stretch of the imagination from what I've shown you before. You develop a submodel for each stream of data, and then you fuse them at the hyperlevel so that fluctuations in, say, fMRI signal should also result in fluctuations in the EEG signal, which in turn signal a change in the decision model. After a long period of data collection, we had data from subjects who gave us both behavior in EEG and behavior in fMRI. In a Bayesian statistical framework, you can actually treat the data that you didn't acquire from some subjects as just missing data. If you then specify the model accordingly, you can actually generate predictions for these missing observations shown here in red. To help the inference structure, though, we need some subjects who perform both tasks so that we know how EEG will typically covary with fMRI. So we recruited a few participants who performed both tasks. They came in 1 week and did the same task in the context of EEG, and then came back a week later to perform the task in the scanner. Now, we could fit a joint model to all of these data and then generate predictions for the red cells or the unobserved data. But that wouldn't be very useful because we have no way to validate the model. So what I'm going to show you today is that we did a sort of cross-validation analysis where we withheld some data and we're going to make predictions for it and then compare the predictions to those withheld data. And so for this example, we can just withhold just EEG data and make predictions for it. Here's an example of what those predictions look like for EEG data. This plot is another posterior predictive density in the space of EEG data, but we can summarize these data by propagating this information onto the scalp for a set of electrodes that were the main focus of our study. Here are the main predictions from the model, and we can compare these predictions against the data that were withheld. At a high level of comparison, we can see the joint model makes reasonably accurate predictions for these withheld data. So now we'll talk about a new approach using a non-parametric linking function along the temporal structure of the data rather than on trial-to-trial parameters themselves. In a more recent line of work, we've been using Gaussian processes as the internal linking function connecting all measurements together. This has been quite powerful because it allows us to directly infer complex linking functions and embedding spaces from data. And so if you're interested in this line of work, Giwan is giving a presentation on this later on in the conference. Recently, principal component analyses have been shown to provide a useful way of constructing embedding spaces to describe the temporal dynamics of brain data. Here we see the results of a PCA on a large set of data with many different tasks, and we see roughly that different principal components account for different neural activation patterns within different tasks. From this result, you can plot the principal components against one another and show how these dynamics play out through time. Here, time is represented by the arrows and isn't reflected on any particular dimension. Our approach is similar, but instead of principal components, we are assuming that the temporal dynamics follow some latent structural form. So first you can specify some generic form for the dynamics, as well as the path and the latent space that those dynamics take. Now we can use the position along this shape at any moment in time to define the state of mind of an observer. So this might describe the valence and arousal of a subject if we were studying emotion, or different attentional states thinking back to our earlier default mode network application. With the embedding space specified, we can now simply connect the streams of data to these latent dynamics. With the connection in place, we can now make specifications about how the data that we might collect from an experiment are connected to the latent dynamics specified at the hyperlevel. In this way, the latent dynamics specify a joint structure from which all measurements we could obtain are jointly constrained. And just to briefly mention that we applied this approach to real data from a motion tracking task, and as you can see here, the model can clearly capture all the essential trends in both behavioral and fMRI data. Beyond that, we get this emergence of latent dynamics from these data streams. This is one example of the type of latent topology that comes out, where each position along this curve is specifying a slightly different state of mind. This provides a unique set of constraints within the data space. If we truly believe that neural and behavioral data provide meaningful information about the mind's computation, then developing ways of integrating the two fields would be a useful endeavor. But we can't hope to directly connect these variables if the purpose of our inquiry is the mind's computations. To do this, we must either develop a theory of the mind, or we must posit latent variables to quantify the degree to which a linking hypothesis is evident. Until we have a grand theory of the brain that we can all agree on, joint modeling is just one convenient way to evaluate how the brain produces behavior. There are many people who have contributed to this line of work. Here are just 6 of them, as well as several really smart people in my lab back in Ohio.